Imagine a leafy herb trusted for centuries in Asian kitchens and medicine cabinets, now labelled a deadly toxin in the West. Science reveals Perilla's true identity is split by a single molecule, condemning some plants and exonerating others. Livestock outbreaks tainted its reputation, but no human has ever been harmed. So why did we abandon a safe, nutritious leaf? The real chemistry behind the controversy starts here. Perilla frutescens is an herb that wears many faces, each shaped by centuries of cultivation and local tradition. In Japan it is known as shiso, usually green-leaved, prized for its aromatic bite and used to brighten sushi or salads. In Korea the same plant takes on the name kaenip, with broad, tender leaves that are often pickled or grilled. Across China it is called zisu, where both green and purple forms appear in medicine and cuisine. These names are not just linguistic quirks, they reflect real differences in the plant itself. Some Perilla varieties have been bred for their leaves, others for their seeds. Leafy types dominate kitchen gardens, valued for their flavour and gentle aroma. Oilseed types, on the other hand, are grown for their tiny, oil-rich seeds, less fragrant, sometimes with a red or purple tint. But not every Perilla is created equal. Beneath the surface, small genetic differences shape what the plant produces. For example, Japanese shiso is typically low in perilla ketone, the compound linked to animal toxicity, while some wild or oilseed strains in China and Korea may contain more of it. These distinctions matter. The perilla on a sushi plate is not the same as the wild mint that sometimes grows in American pastures. Knowing the difference between culinary cultivars and their wild cousins is the first step toward understanding why this plant has been both trusted and feared. Inside a quiet laboratory, a phytochemist prepares a sample of Perilla leaf for analysis. The machine of choice is gas chromatography mass spectrometry, abbreviated GC-MS, a tool that separates and identifies the plant's chemical components with precision. The sample is injected and soon the chromatogram reveals its story. Two distinct peaks appear at different retention times. The first, sharp and prominent, belongs to perillaldehyde, an aromatic aldehyde that defines the flavour of culinary shiso and kaenip. Its molecular structure features a reactive carbonyl group bonded to a hydrogen, giving rise to the familiar citrus mint scent. The second peak, appearing later, signals the presence of perilla ketone. Unlike the aldehyde, this molecule carries a ketone group, an oxygen double bonded to a carbon flanked by two other carbons. This small structural change is enough to alter its biological effects. In the chromatogram, culinary cultivars show a dominant perillaldehyde peak, while wild or oilseed types sometimes reveal a strong perilla ketone spike. The GCMS evidence leaves no doubt. Not all perilla plants are chemically alike. The laboratory data make the invisible visible, tracing the line between aroma and risk. These molecular fingerprints lay the groundwork for understanding why some animals, but not humans, face danger from the wrong kind of perilla. A veterinarian kneels beside a motionless heifer, her breath shallow and laboured. The pasture is dotted with clumps of purple-stemmed perilla mint, the weed now infamous among local ranchers. In the fall of 2018, 3 separate farms in West Virginia lost a total of 35 cows within days. The animals had grazed on perilla mint after summer grasses ran short. Necropsies revealed lungs swollen with fluid, a textbook sign of atypical interstitial pneumonia. The cause was perilla ketone, concentrated in the plant's seeds and flowers late in the season. The pattern is grimly familiar. In October 2023, another case surfaced. This time, a 5th cow in 6 years on the same property showed sudden respiratory distress. Previous incidents on that farm had ended quickly. 4 cows died within 48 hours, each episode traced back to the same weed. Pasture inspections confirmed dense stands of perilla mint, thriving where the grass had thinned. Veterinary reports from across the southeastern United States echo these findings. Outbreaks cluster in late summer and early fall when forage is scarce and perilla mint is at its most toxic. Feeding trials have shown that just 5 pounds of seed-stage perilla hay can kill a young calf in 3 days. In herds caught unaware, the losses mount quickly. The numbers are small compared to national cattle populations, but for the families involved, each loss is a blow. These cases give a face to the risk, a reminder that for livestock, the wrong leaf at the wrong time can be fatal. Inside the ruminant digestive tract, the story of Perilla ketone takes a dangerous turn. Cattle and sheep possess a multi-chambered stomach, with the rumen acting as a fermentation vat. When these animals graze on wild Perilla mint loaded with Perilla ketone, gut microbes begin to break down the compound. This process transforms Perilla ketone into a reactive metabolite that enters the bloodstream and travels straight to the lungs. Their pulmonary enzymes, particularly cytochrome P450s, further activate the toxin. The result is a sudden increase in the permeability of lung capillaries, flooding the air sacs with fluid and making it nearly impossible for the animal to breathe. Veterinary pathologists regularly find lungs swollen and heavy, filled with frothy fluid, a classic sign of atypical interstitial pneumonia. The difference between species is stark. Ruminants, with their unique fermentation system, are especially vulnerable. Experimental trials have shown that as little as 5 pounds of Perilla hay in the seed stage can kill a young calf within days. Sheep too succumb at doses of 15 to 25 milligrams of Perilla ketone per kilogram of body weight. Horses and goats are not immune, but cattle are the most frequent victims, especially when pastures are overgrazed and alternative forage is scarce. Humans, by contrast, do not have a rumen. The small amounts of Perilla consumed in traditional diets, whether as fresh leaves, pickles or oil, are digested and metabolized in the liver, bypassing the dangerous bioactivation pathway seen in livestock. No documented cases of human poisoning from Perilla ketone appear in medical literature, despite centuries of culinary and medicinal use across Asia. The risk, it turns out, is a matter of biology, dose and context, not an inherent flaw in the plant itself. In a bustling kitchen in Seoul, a chef farmer lays out a basket of freshly harvested kainip. The leaves are washed, stacked and brushed with a marinade of soy sauce, garlic and sesame oil, then pressed into jars for pickling, a process repeated in Korean homes for generations. At the morning market in Kyoto, green shiso leaves are bundled for sale, destined to crown plates of sashimi or to be battered and fried as tempura. In rural Sichuan, sprigs of zisu are simmered with fish believed to ease digestion and ward off foodborne illness. These scenes are not relics of the past, they are part of daily life. References to Perilla appear in the Ben Cao Gang Mu, the 16th century Chinese pharmacopoeia, where Li Shizhen described the leaf as a remedy for cough, nausea, and even fish poisoning. Whether as a garnish, a pickle, or a medicinal tea, Perilla's presence in Asian kitchens and clinics tells a story of trust built over centuries. The hands that prepare it, from market stalls to family tables, treat the leaf with respect but not with fear. Perilla seed oil stands out as one of the richest plant sources of omega-3, with alpha-linolenic acid making up between 50 and 65% of its total fatty acids. This rivals and sometimes even exceeds the alpha-linolenic acid content found in flaxseed oil. Nutrition scientists have pointed to this profile as a key reason for Perilla's reputation as a heart-healthy oil in parts of Asia. Yet for all its promise, Perilla never found a foothold in the global edible oil market. After the Second World War, agricultural policy and economics pushed farmers toward monocultures of corn and soybeans. These crops benefited from subsidies, research investment, and global trade agreements, while Perilla remained a minor, mostly local crop. As soy and corn acreage soared, reaching over 80 million acres each in the United States by the 2000s, Perilla's cultivation dwindled to small plots and backyard gardens. Today, most people outside East Asia have never tasted the leaf or its oil. Chef farmers who champion Perilla often reflect on what was lost. A diverse, nutrient-rich food that could have played a bigger role in modern diets, if not for the economic forces that pushed it aside. Today, millions consume Perilla leaf safely, while outdated myths still shape Western food policy. As nutrition science advances, what we exclude from our plates matters as much as what we include. Sometimes the real risk is letting fear, not evidence, dictate what we eat. Share your thoughts below.